Hi everyone. So today I really want to talk about sleep physiology because I feel it is a very important aspect of physiology and medicine that we as anesthesiologists should focus on more. Because essentially, if you think about general anesthesia, superficially at least, it kind of looks like the person is asleep, right? We are putting and even in layman's terms, we generally talk about general anesthesia as putting a patient to sleep. So I think it is very vital and important to have a basic understanding of how sleep works, what are the various sleep centers and arousal promoting centers that are there in the brain. So let's start off by actually discussing what is sleep. Now, sleep, as we all know, is a very important physiological process that is necessary for survival. In fact, it was found in several studies, experimental studies on rats that were devoid of sleep, they died within 1 to 2 weeks, right? Which is the same amount that is seen when they are devoid of any nutrition. So, starvation will also result in death within 1 to 2 weeks, and same is with sleep. If there is no sleep, the experimental rats, died within 1 to 2 weeks. So, this kind of hints towards the importance of sleep. Sleep is very important for a lot of regenerative processes that are happening in the body. Even in healthcare and medicine also, sleep is very important because a lot of times we encounter road traffic accidents or motor vehicle accidents where the person essentially reported being asleep at the wheel. There are a lot of psychiatric disorders like depression, schizophrenia, etc., that are associated with a lot of sleep disturbances. There are certain disorders specifically associated with sleep, such as narcolepsy, right? Which is essentially when the person suddenly goes into deep sleep. And then for anesthesiologists, a very important aspect of sleep pathophysiology is the development of obstructive sleep apnea. So it has very important perioperative implications for anesthesiologists. Now, broadly speaking, if you're talking about sleep, there are various stages of sleep, but sleep has been divided principally into REM sleep and non REM sleep. Right. And each sleep cycle is around 80 to 120 minutes long. So I'm not talking about the whole of sleep architecture, I'm just talking about one cycle within that entirety of sleep architecture. A complete night's worth of sleep will have 5, 6, 7 of such sleep cycles. So each sleep cycle is going to be about 80 to 120 minutes long, out of which about 60 to 90 minutes are spent in non REM sleep, and about 10 to 30 minutes are spent in the REM sleep. Now, what do we mean by REM and non REM sleep? So, essentially, this portion, REM, actually refers to rapid eye movement. And it is a characteristic feature of REM type of sleep, which obviously gives the stage its name as well. Now, REM sleep or non REM sleep, where we spend most of our time during a sleep cycle, we spend it in the non REM sleep. It is characterized by essentially waxing and waning muscle tone. There is a decrease in body temperature. There is a decrease in the heart rate. The respiration is usually deep and regular. If you look at the EEG patterns in the non REM sleep, this is characterized by slow waves of EEG. We will also talk about the various EEG signals that are seen in various stages of sleep. And in the deep non REM sleep, you are also going to see delta waves on the EEG. And what about the REM sleep? So REM sleep is marked essentially by a high cortical activity. If you take a look at the EEG during REM sleep, you will see that the EEG kind of resembles the waking state, because in the waking state you are going to have high frequency, low amplitude beta signals, whereas in REM sleep as well there is high cortical activity even though the patient is asleep. And that is why it is said that sleep is not simply an absence of activity, because there is so much more going on inside the brain that is not evident superficially. Therefore, you cannot just classify sleep as an absence of activity, because it is so much more. REM sleep being characterized by high cortical activity. Characteristically, also, there is marked muscle atonia. So, essentially, all of the skeletal muscles except for the extraocular muscles, they are in an atonic state. They are essentially paralyzed. The person, when they are in the REM stage, they are essentially paralyzed, except for obviously the extraocular muscle, which are obviously showing rapid eye movements, giving the stage its name. This stage is also associated with penile and clitoral engorgement, and usually dreams happen during the REM stage of sleep, even though the person can still dream during the non-REM portion of their sleep, but most of the dreams that happen during the REM part of the sleep. Now, there is another very peculiar feature of REM sleep, is that there is essentially an operation of a system that is going to suppress the motor activity. Suppression of motor activity, which is obviously going to result in muscle atonia, which I previously mentioned. Now, why is this important? This is important because it prevents the person from acting out their dream. You can very well imagine, if a person is dreaming, there is high cortical activity. If all of those impulses will be transmitted to the skeletal muscles, there will be erratic movements of the limbs. The person might fall, the person might injure themselves. So, in order to prevent the person from acting out their dream, the brain has an operation of system that suppresses this motor activity. However, there are occasional breaks in the system which is responsible for that sudden jerk that you feel during your sleep, right? A sudden feeling of falling down the stairs or something, right? So this is essentially your two stages of sleep, your non-REM sleep and your REM sleep. It has been found that all of the organisms throughout the all of the animals especially, they show a certain kind of sleep, not exactly the kind of sleep that would be seen in mammals, because mammals and birds are the only organisms that show this distinction of non-REM and REM sleep. Rest all of the organisms, reptiles and whatnot, amphibians and fishes, they have periodic or rather they have these specific periods of relative inactivity followed by high activity. So the relative inactivity, kind of, you could say that is a sleep stage, whereas high activity would be associated with wakefulness, but they do not have these characteristic REM and non-REM stages of sleep. So, as I've already mentioned, this structured temporal order of different stages of sleep is known as sleep architecture. Right? And this sleep architecture can be visualized by something which is known as a hypnogram. So, what is a hypnogram? It is simply a graphical representation of various stages of sleep with respect to time. Right, so essentially, if this is your y-axis, or x-axis, and this is your y-axis, the x-axis is going to show your time, essentially the time spent in sleep. Let us suppose this is 10 p.m. and this is 6 a.m. So this essentially represents one full sleep architecture comprised of various sleep cycles. Now, each sleep cycle is going to start with wakefulness, then the person becomes drowsy, and then they enter the non-REM portion of their sleep, right? So this is the non-REM portion of their sleep. Then they will have relatively short period of REM activity followed by a brief arousal, followed again by non-REM sleep, then REM sleep, then a brief arousal, and so on so forth. It continues until the time the person is ready to wake up. So this is known as a hypnogram, right? Now, if we talk about the various EEG waves that can be seen, now EEG waves obviously measure cortical electrical impulses of the brain through scalp electrodes. Now, in the waking stage, when the eyes are open, eyes will be open, the EEG is mostly dominated by beta activity. Right, so beta waves are waves which have a frequency of more than 13 hertz. So in most waking states, the EEG will be dominated by beta activity. Then you also have alpha waves. Now, alpha waves are lower in frequency than beta waves and higher in amplitude. Usually, the frequency ranges from 7 to 13 hertz. Then there are theta waves, which essentially are going to range from 4 to 7 hertz. And then there are the very low frequency, high amplitude waves called the delta waves, which will be less than 4 hertz. Now, theta and delta waves will only be seen in sleep, and they will not be seen in waking stage, especially in adults. In children, there might be occasional instances where you could see these waves during wakefulness, but in adults, theta and delta waves are seen only in sleep. And there is something known as alpha to theta transition. And this transition actually is quite sudden. And this marks the stage 1 non-REM sleep. So when the EEG pattern suddenly changes from alpha waves to theta waves, then we say that the person has now fallen asleep, right? And then you also have stage 2 sleep. Now, stage 2 non-REM sleep is characterized by essentially two types of structures in the EEG. First, we have the spindles, which are essentially alternating areas of the EEG with increasing and decreasing amplitudes. And then you also have sudden sharp complexes, which are known as K complexes. So essentially, what are K complexes? What is the reason for K complexes? Although the exact reason is not really known, it has been hypothesized that complexes occur due to activation of certain brainstem and your subcortical essentially areas in the brain. So they get activated. Why do they get activated? They get activated because they are processing certain unexpected sensory stimuli. Unexpected sensory stimuli. Most commonly auditory stimuli. So these, even though the person might be asleep, but they would receive certain auditory stimuli, and sometimes a very loud noise has to be processed because it might mean that there is some potential danger which would require cortical arousal and a return to consciousness. These are protective mechanisms to protect the animal from potential danger. So, in order to process these unexpected sensory stimuli, these brainstem and subcortical areas get activated, which translate in the EEG as K complexes. That is one of the proposed mechanisms of why K complexes occur. And it has been found in various studies that comatose patients who show K complexes in their EEG have a better prognosis than patients without any evoked K complexes. So that's quite an interesting thing to note, essentially. Right, so this is essentially what is your basic thing about sleep, how it what are the various stages of sleep, and what are the various waves that you can appreciate in the sleep cycle. Now I really want to talk about the neuroanatomy behind sleep and arousal. Okay, so talking about the neuroanatomy behind sleep and arousal, I want to keep it really, really simple. So, see, essentially, whether the patient is asleep or if they are awake, these are the two states that we are concerned with. Whether the patient is in any one of these states will be decided by a balance, right? Balance between the sleep promoting regions in the brain and the arousal promoting regions in the brain. Now, there are two ways we can go about looking at the neurophysiology of sleep. First, we will talk about the various centers that are involved, and then I will talk about the two most important regulatory mechanisms behind how sleep works. So, if we talk about the arousal centers, what are the arousal centers? Actually, before we even go about talking about these arousal and sleep promoting centers, we have to have a basic understanding of the neuroanatomy of the uh, brain, and especially the brain stem. So let us imagine, for example, this would be your corpus callosum, right? This will be your anterior commissure of the corpus callosum. This would be here the habenular commissure that is said to be here. Below the habenular commissure, you have the pineal gland. Below the pineal gland, you have the posterior commissure, right? Similarly, below the anterior commissure, you have something known as the lamina terminalis. Then you will have the optic chiasma. Then you have the hypothalamus. This entire region is the hypothalamus. Here you will have the tuber cinereum with the infundibulum and the pituitary. Behind that you will have the mammillary body, and then you will have the brainstem, right? So brainstem mein what all structures do you have? You have the midbrain. So the midbrain is characterized by the superior and the inferior colliculi. Then you have the pons, then you have the medulla, and finally the spinal cord, right? And here you are obviously also going to have your cerebellum. And you will also have the fourth ventricle that is present here. This is the cerebral aqueduct of Sylvius, right? So it kind of goes like this. And here also you are going to have the fornix. So if I just label this out entirely, it will become a lot more clearer. So this is the corpus callosum. This would be the anterior commissure. This is the lamina terminalis. The optic chiasma. This is your pituitary. These are the mammillary bodies. This is the septum pellucidum. This is the fornix. The habenular commissure. The pineal gland, which obviously releases melatonin. This is the posterior commissure. The superior colliculus, the inferior colliculus. So this region is the midbrain. This is the pons. This is the medulla. The cerebellum, the fourth ventricle, the aqueduct of Sylvius. Right, so this is how it goes. Naturally, this is a sagittal section of the brain. And obviously, this portion will be the thalamus. Very, very important structure in the diencephalon. And this is the hypothalamic sulcus, and this is the hypothalamus. So once we know this basic architecture of the diencephalon and the brainstem, we can move ahead to naming the various nuclei that are involved in the arousal. Now, the various nuclei that are involved in the arousal are bunched together into something known as the AAS. This is also known as the ascending arousal system. So ascending arousal system are a bunch of nuclei that have various connections. So what are the important ones, right? So the important ones that you need to keep in mind are, so first we have the locus ceruleus. Now locus ceruleus, where is it located first of all? So it is located in the pons, right? So it is located somewhere around here. And what does it release? It releases noradrenaline and it promotes arousal. Naturally, all of these things are going to promote arousal. Now, locus ceruleus is going to have various connections that are going to go into the thalamus. They also go into the basal forebrain, and they also have direct connections to the higher cortical centers. So, essentially, it is going to stimulate the cortex and promote wakefulness, right? So, this is quite important. Locus ceruleus. Another pair of nuclei that you need to know are the pedunculopontine tegmentum and the latero-dorsal tegmentum. So they are located just above and dorsal to the locus ceruleus, somewhere around here. Now they are also obviously located in the pons only. And they release acetylcholine. So they are cholinergic in nature, again promoting arousal due to various connections. The third center that you need to keep in mind is a very important one is the TMN or the tubulomammillary nucleus. Now this is the nucleus that is actually located in the hypothalamus. It is not located in the brainstem, it is located in the hypothalamus. So it is located in the hypothalamus. And what does it release? It releases histamine. Principally, it releases histamine. Then fourth center that you also need to keep in mind is something known as the VTA, also known as the ventral tegmental area. Now this is located here in the midbrain. So this is located in the midbrain and it releases dopamine and it also promotes arousal. And finally, a very, very important, apart from all of these structures, a very important structure for arousal, center in the lateral hypothalamus, is known as a group of neurons called the orexinergic neurons. Now, orexinergic neurons are a special group of neurons that release, you guessed it, substances which are known as orexins. Orexins are also known as hypocretins. And there are two types. We have orexin A and orexin B, or hypocretin 1 and hypocretin 2. Now this is very, very important in the flip-flop switch. of sleep-wake cycle, which I will talk about in just a moment. This flip-flop switch is very, very important, and the orexinergic neurons located in the hypothalamus are responsible for this flip-flop switch between sleep state and the arousal state. So these are the various nuclei that are part of the ascending arousal system. Now, why is it called ascending? Because as you can see, these arousal signals or stimuli are originating in the brain stem, right? And then they are traveling up towards the cortex. They are ascending towards the cortex and causing the brain to become aroused and the person to be in the wakefulness state. In addition to keeping the cortex aroused, another job of the ascending arousal system is to reciprocally inhibit the sleep promoting areas. Now, there is just one sleep promoting area that you need to know, which is the most important. I think I forgot one other center in the arousal promoting region, and that is the pontine reticular formation. Right, so and also the dorsal median raphe nuclei. So pontine reticular formation naturally will be the area in the pons, the reticular formation of the pons, and this actually releases GABA. Now this is very, very interesting because in all the other centers of the central nervous system, GABA is going to act as an inhibitory neurotransmitter. We know this from MBBS, right? Glutamate is the excitatory neurotransmitter, GABA is the inhibitory neurotransmitter. But within the pontine reticular formation, there is GABA induced arousal, which is very, very interesting to note. So that serves to tell us that there are no hard and fast rules in biology, right? Because in pontine reticular formation, GABA serves to stimulate arousal rather than sleepiness. Anyways, and the dorsal medial raphe nuclei, they release serotonin, which also serves to keep the animal aroused. So these are all the ascending arousal system. Then we have the sleep promoting area, and the only most important one that you need to know is the VLPO. Right, so this is located in the anterior hypothalamus essentially. So this stands for ventrolateral preoptic nucleus. So if you take a look or if you have studied the structure of hypothalamus is actually divided into a lot of different zones. For example, this is the hypothalamus. It is divided into a lot of zones. I don't remember the exact names of them, but they are supraoptic, there is arcuate zone, then there is something zone. So there are different zones in the hypothalamus. So within the hypothalamus, there is this center known as the VLPO or ventrolateral preoptic nucleus, which is principally responsible for promoting sleep. Now this is going to release two principal neurotransmitters. First is the GABA, which as expected acts as the negative or inhibitory neurotransmitter, and it also releases galanin. So these are the two neurotransmitters which are released by VLPO. So these arousal centers, when the patient is in a waking state, they are going to arouse the cortex, but they are also simultaneously going to inhibit the VLPO so that the animal does not go to sleep. Similarly, during the sleeping state, the VLPO is very, very active and it serves to suppress the arousal promoting area so the animal does not wake up. Right? So now, earlier I mentioned about the regulatory mechanisms of sleep-wake cycle. So how does sleep-wake cycle gets regulated? So when we talk about regulation, there can be slow changes in regulation and there can be fast changes in regulation. The fast changes in regulation I have already mentioned. It is the flip-flop switch. Right? Which will instantly put an animal to sleep. If this switch is in the on state, then the animal gets active and aroused. If it is in the off switch, the animal will fall asleep. The slow changes or the slow mechanisms of regulating the sleep wake cycle are further divided into two types. First, we have the homeostatic drive, and we have the circadian rhythm. So, what do we understand by homeostatic drive essentially? Right? What is it? So, according to homeostatic drive, it says that throughout the time the animal or the person is awake, some substance or substances keeps on building inside the body. Its concentration successively keeps on rising and rising and rising up until a point the concentration of that substance becomes so much that it activates the sleep promoting area, and the animal wants to fall asleep and becomes drowsy and slowly gradually drifts off to sleep. That is what we call the homeostatic drive. Now, we don't exactly know what those substances are, but a very good candidate for homeostatic drive is adenosine. So, throughout the day or throughout the waking hours, the adenosine concentration keeps on building and building and building. Also, a very interesting thing to notice that the receptors with which adenosine interacts in the central nervous system are antagonized by caffeine. And therefore, caffeine blocks the central nervous system effects of adenosine. That is why when we drink coffee, we feel less tired and more awake, because it is blocking this homeostatic drive. Second slow regulatory mechanism is the circadian rhythm. So, circadian rhythm is a very, very important feature wherein the person or the animal is going to show not just behavioral changes but also various physiological and metabolic changes within the body in response to external light and dark stimuli. For example, during the daytime, there is more of light, ambient light, and that is going to change the behavior of the animal. For example, for diurnal animals, they will be more active up and about foraging during the daytime, and they will be less active during the night. This circadian rhythm is flipped in nocturnal animals, where they are more active during the night and less active during the day. So it is not just the behavior that is affected by circadian rhythm, it is also the internal metabolic and physiological processes that are occurring. For example, the secretion of melatonin, secretion of catecholamines, cortisol. So all of these, the various cardiovascular changes that happen in the basal heart rate, the respiratory rate, secretion of various hormones. So all of those things are affected by circadian rhythm, and virtually all animals have some amount of circadian rhythm associated with them. Like I already mentioned, except for mammals and birds, most animals do not have non-REM and REM type of sleep, but they do have circadian rhythms. So circadian rhythm was established very early on during animal evolution. So how does it work? So circadian rhythm actually works on the principle of a master clock. Right, so within our brains there is a master clock that actually knows when it is the daytime, when it is the night. So what is that master clock within the human brain? So it is known as the suprachiasmatic nucleus, and it is actually very easy to relate with what we already know. Supra means above, chiasmatic relating to the optic chiasma. So naturally, the suprachiasmatic nucleus is situated in the hypothalamus just above the optic chiasma. And because of its proximity to optic chiasma, it is at a very good position to get signals about visual information and light intensity from the visual pathway, because it is so closely attached or related to optic chiasma. So it is going to receive information from the retina. So I'm just going to draw a very simple flowchart type of a diagram demonstrating how this circadian rhythm works and how it integrates with the flip-flop mechanisms. So let us suppose this is the suprachiasmatic nucleus. It is going to receive information from the retina about the light intensity. Now, in turn, the suprachiasmatic nucleus is going to stimulate the dorsomedial hypothalamus. Right? And in turn, in addition to stimulating the dorsomedial hypothalamus, it also is going to inhibit paraventricular hypothalamus or lateral hypothalamus. We don't really know the exact center, but it inhibits this area of the hypothalamus. This area in turn is responsible for the stimulation of the lateral horn of the spinal cord, especially in the segments T1 to T2. Now, if you remember from neuroanatomy, this is the spinal cord, the gray matter, the white matter. So you have the anterior horn, the dorsal horn, and there are lateral horns as well, where you have the origin of the preganglionic sympathetic fibers or autonomic fibers, right? So these originate from the lateral horn. They travel in the this dorsal root, and then they through white and gray rami communicants, they are attached to the sympathetic chain or sympathetic ganglia, which are situated next to the spinal cord in the paravertebral gutters. So from here, the lateral, these paraventricular hypothalamic cells are going to stimulate the lateral horn, through which the autonomic fibers will originate and go towards the travel within the sympathetic chain and go towards the superior cervical ganglion. And from the superior cervical ganglion, there will be stimulation of the pineal gland, which in turn releases melatonin, and melatonin in turn has an inhibitory effect on the suprachiasmatic nucleus. So what does this dorsomedial hypothalamus do? It is essentially going to stimulate your lateral hypothalamus or the orexinergic neuron that I talked about earlier, and other arousal centers. So what is going to happen is, let us suppose it is the daytime. There is ample information coming in from the retina about the light source. It is going to stimulate the suprachiasmatic nucleus. Once the suprachiasmatic nucleus is stimulated, the inhibition on the paraventricular hypothalamus increases. That means this stimulation of pineal gland is reduced. So during the daytime, pineal gland is not very much stimulated. Rather, it is suppressed, and therefore there is very little amount of melatonin being released during the daytime. If there is very little amount of melatonin being produced, this inhibition over the suprachiasmatic nucleus is also not there and is lifted. Therefore, suprachiasmatic nucleus acts a lot during the daytime, which serves to stimulate more and more the dorsomedial hypothalamus, which in turn activates the various arousal centers, right? And this is how the circadian rhythm functions. During the night time, because there is not much signals coming in from the retina, the suprachiasmatic inhibition of the paraventricular hypothalamus is lifted, and therefore the pineal gland is stimulated a lot more, and a lot more melatonin is formed, which further suppresses the suprachiasmatic nucleus, which serves to decrease the stimulation of dorsomedial hypothalamus, and the arousal centers are not stimulated, and the animal falls to sleep. So this is how the circadian rhythm functions. So changes in sleep behavior in response to external light and dark stimuli. But I also talked about a very important, and same with homeostatic drive also. As the homeostatic drive keeps on building up, the suprachiasmatic is suppressed more and more, and the animal eventually falls to sleep. But what about the flip-flop switch? So now comes the flip-flop switch. So here we have the tubulomammillary nucleus, which I already mentioned. This releases histamine, but it also releases GABA. And here we have the VLPO, which releases obviously GABA. Now we know that GABA has an inhibitory influence. So this VLPO inhibits the tubulomammillary nucleus, whereas the GABA, which is secreted by tubulomammillary nucleus, also reciprocally inhibits the VLPO nucleus, not the histamine, but the GABA that is released by tubulomammillary nucleus. So they reflexively inhibit each other. But which portion of this equation will be more active will be dependent on the external influence by the orexinergic neurons, right? So during the daytime, what happens, there is activation of dorsomedial hypothalamus, which leads to more activation of the orexinergic neurons, which serve to stimulate the tubulomammillary nucleus. So as the tubulomammillary nucleus is stimulated, more and more histamine is released, which will serve to arouse the patient or the animal. And it also serves to increase GABA production from tubulomammillary nucleus, which serves to inhibit the sleep promoting VLPO region. And thus VLPO is not active during the daytime, and the animal is quite awake. But as soon as the night rolls around and the orexinergic stimulation falls, this GABA production by tubulomammillary nucleus also falls, and this side of the equation now becomes more dominant, and VLPO becomes active and serves to suppress the tubulomammillary nucleus, and the animal goes to sleep. In fact, it has been found that in narcolepsy, there is a deficiency of these orexinergic neurons. Due to autoimmune destruction of orexinergic neurons, there is lack of orexinergic neurons. Therefore, this flip-flop switch is faulty, and the person can anytime, their VLPO center can take over, and the person can go into a deep sleep at any point of time. Right, so this is the basic neuroanatomy that you need to have in mind when you're talking about sleep and arousal and sleep wake cycle, because understanding this aspect of sleep physiology is very important, because if we understand this, then only we can kind of understand how general anesthetics, both IV and volatile anesthetics, serve to produce unconsciousness in the patient. Okay, so now that we know about the basic neurophysiology behind sleep, now we can just briefly focus on how anesthetics influence the sleep architecture, right? So even though, like I mentioned previously at the start of this talk, is that general anesthesia might seem similar to, in certain ways, similar to just a state of sleep. However, there are certain key differences between the state of general anesthesia and the state of sleep. For example, sleep, a very, very important aspect of sleep is that it shows spontaneous generation and termination. Right? So a person will by themselves go to sleep and then wake up. So there is spontaneous generation and termination. There is ready reversibility of the sleep state by noxious stimuli. So if you pinch someone who's sleeping, they will naturally wake up. So there is ready reversibility. And obviously there is homeostatic regulation that is intact during natural sleep. All of these things are not found in general anesthesia, right? General anesthesia has to be induced. It is a state of induced loss of consciousness, and it is not spontaneously reversible, right? There has to be some amount of drug elimination that has to occur within the body before the individuals can be aroused. If you take a look at the EEG, then you will see even more marked differences between general anesthesia and sleep. There are similarities between the EEG patterns of the slow wave sleep and anesthesia induced consciousness, but the EEG patterns during sleep and anesthesia are fundamentally different, right, with different frequencies and types of activation. Also, it has been found, if you're recording the EEG and the person is being administered general anesthesia, you will find that the level of consciousness continuously decreases from a fully awake state through various intermediate stages of reduced responses to external stimuli to a completely unresponsive state. So there is a shift from one stage to other. There is a gradual decrease in response till the person is completely unrousable and unresponsive. This is in contrast. If you remember, I talked about the sharp alpha to theta transition in the EEG of natural sleep. That is when we say that the person has entered stage 1 of non-REM sleep. So this is in contrast to the sharp alpha theta EEG transitions, which are seen in patients who proceed from wakefulness to sleep without any transient stages. Right, so these are the few differences that you need to know between general anesthesia and sleep, that even though they might look similar, they are not in fact similar. However, it has been proposed. See, ultimately, nobody really knows how general anesthetics depress consciousness. There have been proposed mechanisms by how general anesthesia might work to produce unconsciousness, but nobody knows for sure. that is how it is. Nobody knows for sure. However, there have been several hypotheses that have been proposed due to various functional MRI studies that have been done on general anesthetics. So it has been found that general anesthetics actually stimulate a lot of the same centers in the VLPO that are activated during sleep, right? And as we all know that VLPO produces a state of sleep, and it also serves to inhibit the tubulomammillary nucleus, which is the flip-flop switch. And this has been found to be the case during general anesthesia as well. So during general anesthesia as well, functional MRI imaging have shown that there is activation of VLPO centers with subsequent inhibition of tubulomammillary nuclei centers. There are a number of problems with this theory, however. First and foremost, it has been found that in experimental rats and mice that have been induced with VLPO lesions, so their VLPO centers are defective, they can still be anesthetized. So if this theory would have been correct, then these rats and mice who did not have functioning VLPO centers should be completely resistant to general anesthesia. But it is found that they can still be anesthetized. Even though they might suffer from profound insomnia, they still can be anesthetized, right? And it has been found that acute lesions of VLPO, they increase the resistance to general anesthesia. They can still be anesthetized, but there is some amount of resistance to general anesthesia. But chronic VLPO lesions, it was found that they had increased sensitivity to isoflurane. So it's quite strange if you think about it. Right? Another problem, so one problem with this theory is this, that even with VLPO lesions, the rats were still being able to be anesthetized. The second problem with this theory is that essentially direct inhibition of the tubulomammillary nucleus in experimental animals resulted in sedation but not unconsciousness. Sedation was there but not unconsciousness. So if this theory would have been correct and the inhibition of tubulomammillary nucleus was being responsible for the state of unconsciousness in general anesthesia, then direct inhibition of tubulomammillary nucleus should also have produced a general anesthesia-like state of unconsciousness, but it has been found that it only produces sedation, right? So these are the few problems with this theory. However, in recent years, there are several studies that have been put forth that say that it is not just the stimulation of VLPO and inhibition of tubulomammillary nucleus that is the underlying mechanism of general anesthetics, but it is also the direct inhibitions of various arousal promoting regions such as locus coeruleus, the pontine reticular formation, the ventral tegmental area, the perifornicial area, the tubulomammillary nuclei, right? So and the basal forebrain, all of these areas are inhibited by general anesthetics, which serves to produce a state of general anesthesia. So the general anesthetics interact through a myriad of networks and interact with the ascending arousal system as well as the VLPO system to produce a sleep-like state, right? But naturally, this is not easily reversible and requires the elimination of drug or the administration of certain other drugs in order to reverse the state of unconsciousness. Another thing to keep in mind is that there are certain interactions between surgery, anesthesia, and the normal sleep architecture. So here we are not talking about the intraoperative period of unconsciousness. We're talking about the general anesthesia has been over now, the patient has woken up. Now, how does the administration of general anesthesia, and by that fact, even the act of surgery, affect the normal sleep architecture of a patient? So, it has been found that when you give GA to the patient during the first post operative night, there is a loss of REM sleep. So, there is no REM sleep or very little REM sleep. And then during the second to fourth post operative day, or post-operative night, there is rebound hypersomnia with increased REM sleep. Right? So general anesthetics do disrupt the normal sleep architecture. Normal sleep architecture. Even with propofol, it was found that in ICU patients who received chronic propofol infusions for sedation, there was no REM sleep. And as you know, REM sleep is very, very important for normal physiological functioning. So propofol infusions had no REM sleep. The patient was sedated, unconscious, but the EEG did not show any REM patterns. Whereas these low dose propofol did allow for some amount of REM sleep. This is in contrast to dexmedetomidine. Dexmed was found to inhibit norepinephrine release from locus coeruleus, and this produces a non-REM like state. And it was found that the sedation and unconsciousness, or the sleep-like state that was produced by Dexmed, most closely resembled physiological sleep than any other sedative that was used, propofol, midazolam, whatever. And that is why there is more and more actually movement towards dexmedetomidine as being used as the principal sedative agent in the ICUs, because a lot of the other sedatives that are used in the ICUs, like benzodiazepines, propofol, opioids and all, they disrupt the normal sleep architecture, whereas Dexmed preserves the normal sleep architecture, right? Also, it has been found that these surgical stimulus itself is a very, very potent disruptor of sleep, and it reduces REM sleep even in the absence of general anesthesia, right? Even if the patient was in regional anesthesia, but the very act of surgery itself does impair REM sleep. So, this is basically how the general anesthetics interact with the sleep centers. We still don't exactly know how it brings about unconsciousness, but we do know that sleep and general anesthesia are not the same thing. They might interact with certain similar centers involved in sleep and arousal, but how do they interact with it, and what essentially results from this interaction is not entirely known. But I hope this talk essentially got you curious about how sleep works, how arousal works, and how general anesthetics interact with various sleep centers, because I personally find neuroanatomy very interesting, and specifically the neuroanatomy of sleep very interesting, because sleep is such an integral part to the human experience, but we virtually know very little about it. So, this is by no means the entirety of the sleep medicine or the anesthetic implications, because a lot of anesthetic implications have to do with obstructive sleep apnea. So that is a whole another, you know, this segment of anesthesia as well, but that I am not going to delve into that part of anesthetic management of obstructive sleep apnea. But I just wanted to talk about, generally talk about sleep and how it is produced, what are the arousal centers, and how general anesthetics kind of interact with sleep centers. So, hopefully, this was somewhat informational. If you want to read more about it, there are two very beautiful chapters given in Miller's. One is about consciousness, memory, and one is about sleep medicine.